Okay, hello and welcome to this next lab class. This we are in this lab class we will be doing the next experiment. So I think we have already completed how many? One, two, three, four, five experiments we have already completed. Okay, so this is the sixth experiment, that is we will be doing pigment dispersion. Two more experiments which are not here in the this virtual lab method. What I will do, I'll be conducting those lab live from class, live from your our laboratory. So that is measurement of pH and conductivity of your fountain solution, dampening solution basically. And another experiment we have, that is your this thing bursting strength tester. So those I will make live videos from your laboratory and we will be doing those experiments. Okay, that I will be releasing those lectures for the laboratory work also. We will be releasing very soon. Now this today's topic, that is determination of how well the pigment has dispersed. Now in our earlier I think the first few classes of your PMS 1, I had talked about your this thing how your printing ink, especially your pasting, is a colloidal solution, right? It is not a solution, but it is colloid essentially, where the dispersed phase is your colorant. In most of the cases, it is pigment, and the dispersed medium is your solvent. So, solvent here is not the volatile nature, as volatile as it is volatile, but it is not as volatile as those used in your liquid ink. So, here the dispersed medium is basically together known as the vehicle in which the pigment or the colorant remains dispersed. So a solid remains dispersed in a liquid. Okay, so that is the basic principle. Now, what is the importance of pigment dispersion? You see, now there are many factors which control pigment dispersion. Solubility of the resin in solvent, that is one of the primary factor. But right now we don't need to get into the details of all that, because this again is topic of the ink class that is under PMS2 in the next semester. But pigment dispersion is very important. What is the meaning of pigment dispersion? It means that each of the pigment particles is uniformly coated by the vehicle and remains uniformly dispersed throughout the ink, and not conglomerate or not coagulate in a specific region. Now you see what is going to happen if the pigment, instead of remaining dispersed evenly throughout your various your vehicle, if it remains coagulated, then what is going to happen? Then the color, if you are printing a solid patch, okay, if you have a solid patch in your plate, only a portion of that solid patch will be printed with that particular color and not all. So that is the primary problem of pigment dispersion. There are other problems also, like one problem known as flocculation. Pigment bleeding happens. So I think I have told you this in the colloid class also. What is pigment flocculation? That means when in contact with the your this thing fountain solution, the pigment particles get preferentially attracted towards the fountain solution rather than the vehicle. That means the vehicle is not capable of uniformly coating the pigment particles or wetting the pigment particles. This problem is known as flocculation. And then what happens? I have shown you one picture also. The entire, suppose there is flocculation or pigment bleeding in yellow ink or cyan ink, then the color of the fountain solution slowly and slowly it will turn yellow or cyan or bluish respectively. Okay, so that is one problem. So this will cause what problem in printing? Tinting problem. That means in the non-image area also you are printing with a yellowish water or a bluish water. That means your non-image area also will get that yellowish tint or bluish tint, and all these things is going to happen. Okay, so that is why you see pigment dispersion, test of pigment dispersion is important. What is the principle behind this test? Let us understand. Principle is there are various types of gauges. Gauges here are solid steel gauges. What happens? It is kind of a grooved, machine grooved. Okay, so one end there is thickness of the groove is high, and another end thickness of the groove is very small. So that what happens? So let us say now in the where the thickness of the groove is very high, then what will happen? There those particles which are having those size, those pigment particles which are having very large size. Large size means the size almost equivalent to the thickness of that groove in that area. Okay, those particles will get stuck in those when I pull it with the help of a blade. Okay, so this is, and then we will be able to identify what is the size of the pigments that are present in the ink. Okay, so let us, without further ado, let us go ahead and proceed with the pigment dispersion experiment. Okay, determination is pigment dispersion, how well pigment has been dispersed in the paste ink. Okay, now this I think I have already explained. So let us talk about the theoretical portion. Yes. Now see, dispersion is used for checking the fineness of grinding of ink. Okay. Grinding means pigment has to be ground or uniformly dispersed. That process is known as grinding. How it is done? Ink is made to pass through triple roll mill. There are other mills also, bead mill, ball mill, etc. I'm not talking about the triple roll mill. That means between multiple rollers, very huge rollers, ink is made to pass. Pre-ground ink, that means non-dispersed ink, mixture of pigment and vehicle, that ink is made to pass through rollers, and with the help of the force and the temperature of the rollers, what happens, the pigment, they were agglomerated or coagulating at first, but due to this pressure, then it is finely ground into smaller particles, and those particles are equivalently or uniformly coated with that help of the vehicle component. Okay, thereby the pigments are not getting attracted to each other, they are preventing themselves from coagulating and remaining uniformly dispersed throughout the ink. Okay, though organic and inorganic pigments are in finely ground dry form, unlike chips, dye chips actually. Okay, fine particles form into aggregates and agglomerates. These agglomerates have to be broken down into their primary particle size, and each particle coated with printing ink vehicle. Now vehicle, you might ask, what is vehicle? Vehicle is a mixture of your resin and the solvent, or distillate you can say. There are other components also. The bigger particle size in offset will result into plate wear. Now suppose what is going to happen if the particle size is large or they are coagulating. Apart from this dispersion, I mean this tinting also, if smaller particles are immediately in contact with the plate and the rollers cylinders are constantly rolling, right, or rotating, these larger particles will wear out those plates, right? This is known as the plate wear out problem. Ink pile up on the plate blanket, and therefore these cylinders will get weared up, or it will be deteriorated in their condition. The pigment particle size or dispersion is measured as fineness of grind gauge with the help of this, consisting of a block of hardened steel into which has been machined a shallow groove. Shallow groove means, I will show you, varying in depth from 0 at one end to 25 microns at the other. Okay, a sample of ink is placed at the deep end, that means where the shallow is larger or deeper, and is drawn forward with towards the shallow, less shallow end, with the help of a steel scraper or a steel blade. Now, observation of any scratches in the ink and where they occur in terms of distance along the gauge gives a measure of the degree of dispersion in the ink sample. So, what is the material in our lab? Also, we don't have the NPIRI gauge. We have the Hegman gauge. Okay, these are different types of gauges that people use. We have the Hegman gauge, like similar example that they have chosen here. Okay, this is the example of a Hegman gauge. What is there? Now you see, this is the shallow end. This is the deeper end. Now the depth of this is nearly 50 micron. Okay, and here it is 0 micron. Okay, so if it is 0 micron here, okay, now what is going to happen? Let us take a look of what the nature of the gauge is like. A hardened steel block approximately 180 millimeter in length. This is 180 millimeter. 63.5 millimeter in width, right, and 12.5 mm in thickness. The top surface of the block shall be ground smooth and flat and shall contain a groove 133.4 millimeter in length. This groove, this entire length of the groove is 133.4, and 12.5 millimeter in width. This is the width, centered in the top of the block. The groove shall be tapered uniformly in depth lengthwise from 50 micron at one end to 0 micron at this particular depth. Okay, now the relationship between depth of the tapered groove and the scale of for fineness of grind is what? Now here you see what is the meaning of this? Now here the depth of the uh, groove is 50 micron, right? Then what is the corresponding result in your Hegman gauge? So this relationship is given. If the depth in here, the depth in micron is 0, right? Then the score or the the value that you are looking for in the Hegman corresponds to 8. Okay, here uh, in when it is here 40, I think 40 micron, the depth of the groove here is 40. Then what is happening? The corresponding Hegman number is 4. So like this it is this comparison chart is there. Now let us take a look at what is the so depending on this theory part and the introductory part, there is some pre-test obviously. Let us take a look. So this is the pre-test, basically some questions in the that you can answer very easily by just going through the theory part and listening to the whatever information I already told you. Then let us take a look at the procedure also. So procedure I will be talking about, but let us take a look at let us go through the procedure here for a minute. Now place the gauge on a flat non-slippery surface and wipe it clean immediately before the test. Place the material means here the pasting, to be tested in the deep end, that is the other end, away from your end of the groove, so that it overflows the groove slightly. Care shall be taken to see that the sample is free of air bubbles. Using both hands, hold the blade perpendicular to the block surface at right angle to the length of the groove. Draw the material down, just scraping of the length of the groove with a uniform deliberate motion. Use sufficient pressure to clean the level surface of the gauge. Immediately read the fineness of as follows. View the gauge from the side so that line of vision is at right angle to the long dimension of the groove. That means if the groove is like this, then your vision should be like this, 90 degree. Hold the gauge in light that will make the pattern readily visible. What happens if the size of the pigment particles is very large? Then as you are scraping the ink from the deeper end to the shallow end, those larger size pigment particles will kind of drag along with the blade, creating these patterns, these lines. Okay. For actual reading, make the angle between the face of the gauge and the line of vision not more than 30, not less than 20. Normally scratches means the pigment particles that are being scraped by the blade, that will cause the scratch. Now imagine how scratches appear. This is the pigment particle, right? And ink is over here. You are trying to scrap the ink with the blade. Now pigment particle being larger, it will block the ink to flow in this particular line, and as you scrap the blade, this larger pigment particle will also come down, and it will cause a line or a particular pattern to appear. Okay. Normally scratches will appear on the surface of the ink due to agglomerates of the pigment particles. That means dispersion has not been done. Pigments are still coagulating. Note the scale interval in which 3 or more scratches commence and record to the nearest scale division. Use a fresh sample of the material each time. Obtain 3 readings in like manner. The first drawdown means this drawing pattern. And reading is preliminary in order to establish proper conditions and to locate the fineness pattern. So multiple times you need to do this with the untrained person. With this known, the second and third readings can be made with minimum time lapse between completion of drawdown and actual reading. Why this is said that immediately after drawdown you should note down the reading? Otherwise, there may be some particles that may drop, some dust particle that may fall, that will that will actually give erroneous readings, or the flow of the ink might be caused, due to which that will also cause erroneous readings. No reading shall be considered for reporting fineness when the time lapse exceeds 10 seconds. So within 10 seconds you need to do this. In case of highly viscous inks, a little medium should be used for bringing the material to workable consistency. There are some metal deco inks which are having very high viscosity. There you should apply some medium. Medium is a vehicle only, without the color, that medium, so that the viscosity comes down a little bit. Now care of the gauge. How care of the gauge should be done? The gauge shall be immediately cleaned after each reading. Use a solvent and soft cloth. Keep the gauge covered at all times when not in use. Do not allow any hard material to come in contact with the gauge surface or the scraper, so that that fine groove that may be destroyed. So let us take a look at how the process is actually done. A YouTube video is also here, so let us take a look. So this is the Hegman gauge basically. This is the spatula or the ink knives using which the ink will be first smoothened out, and then ink various properties will be tested basically. Kerosene is the solvent to wipe, clean the excess amount of ink. So this they are testing with the help of the magenta ink over here. So you see, when the ink is in the container, it has developed this viscous nature, you know. So it needs to be broken down. Otherwise, the scrapping process will not be that smooth. So they will slowly and steadily start to mix it a little bit. The two inks they are testing. So why, you may ask, that there are two, you can say, these grooves that are drawn. One is for the standard ink, one is for the reference ink, so that you can compare both the inks side by side. Two grooves, as you can see, one on the left, one on the right. That is why we have a little bit wider Hegman gauge. See, here it is written in the along the height of the Hegman gauge, but in our case it is written sidewise only. On one side there is the Hegman scale, another side there is the micron scale. Normally 10 micron is a good reading, good dispersion. If there is 10 micron for pasting, normal purpose, it is good dispersion. So two inks will be applied along the two grooves. One ink applied on the left, another ink applied on the right. Maybe one is reference. Actually, in the industry, one is reference, another is the batch, which the ink manufacturer has just made to test the dispersion. This is the steel scraper, and this is how they are scrapping. See how they are holding it 90° and then scrapping the surface. Okay, now immediately within 10 seconds they are now looking. Now take a look nicely that how, although actually one problem here, you see, this is your very much kind of well dispersed ink. Finally manufacturing is very, very well dispersed, but still you can check the dispersion quantity. See where these patterns, these lines are coming from. Very, very fine. I think not able. Yeah, 10. So as I told you, that finished ink, it is normally 10 micron dispersion. Very good. Okay, so this is the procedure. Let us do the simulation also. So let us click on the apparatus. What is required? Ink is required, Hegman gauge is required, and scraper is required. So let us click on the scraper first. This process we don't do. This is not actually the process of applying ink. We normally apply the ink with the help of this thing only, with the help of the knives. So here they have shown they are dipping the scraper on the ink and then applying it on the scale, Hegman gauge, but that is not the process actually. First we, like you saw in the video, that was the correct way. First you put two inks in the on these grooves, deeper edge of the groove with the help of knife, and then you pull it with the help of the scraper knife. Yes. Let us see the result. See what is the result. Let us take a look. What is the result? If you take a look at the result, you will notice one thing. These type of patterns, as you can see, these type of patterns that can be found. What happens, as I already told you, why are these straight line, these not straight line, but these lines are coming, these patterns are coming? Why? Because you see, there are pigment agglomerates or pigment particles of the size, agglomerates of the size, maybe here, how much? Maybe 9, 10 or 9 microns, right? From here actually it is coming, right? That means what? That means when you are this particular, when you have reached these pigment particles, and when these pigment particles have reached these grooves where the length, where the depth of the groove is that much, 9 micron basically, or 10 micron, then it is sitting on that groove or that particular end. And when you are trying to scrap the metal, when you are trying to scrap the uh, ink downward, then it is kind of creating this type of a pattern, this type of pattern, right? So that is how you can test basically, and this is a better, closer look of the experiment. Okay. Yes, so here you can see that in the micron scale, as you can see, the micron dispersion in micron of the ink, particular ink, is near about 12 micron. Okay, so you will write down in your lab report that instead of three readings, normally they have given for three reading, but instead of three reading, you will simply write down 12 micron in the reading. One reading is sufficient. Okay, so is the agglomerate size in given ink sample is 12 micron. Okay, and after that, as usual, there is a post test. There are some questions, 3 questions, easy questions, and simply answer the questions. And in the reference section also, the IS standards referring, which have been referred while conducting the particular test, has also been referred. Okay, so thank you for listening to this virtual lab process. Okay.